now moving from imaging to non-invasive tests, and it's a great pleasure to announce John Blume, who has made the long way from San Diego, California to Heidelberg to tell us his new exciting results about the serum markers.
Great, and thank you very much. And my thanks to the organizers, Dr. Marr, for organizing this session. I plan on learning a lot over the next 2 days. I'm sure that you all will as well too. My goal, as you can see from the name of the company, and I think I'm one of the only companies that's here, and for that I apologize, but we actually do some nice science at the same time, is to focus on proteins as potential markers for disease, and specifically for colorectal cancer and advanced adenoma detection. It wouldn't be a company talk if I didn't tell you a little bit about our origins. The company was founded by Dr. Danny Hillis and Dr. David Agus, who, if you're familiar with an American movie called The Odd Couple, represent an odd couple in science, Walter Matthau and Jack Lemmon. Danny Hillis, a computer scientist who thinks a lot about systems, a lot about handling information, satellite image processing, telecommunications processing, credit card fraud transaction processing, trying to pull signal from the noise. David Agus has always focused on molecular medicine and oncology. He was at UCLA and is now at USC, and has really been at the forefront of trying to get these new technologies, sequencing, microarrays, proteomics, metabolomics, to really fulfill their promise to be able to detect things that are going on. They both fundamentally believe that the technologies have been there, they just really haven't been applied in the right way. And perhaps partly it's because of the questions that we've been asking. And so the company was formed to try and explore this and see whether or not thinking about things in a new way would actually help us get to some of these indications. This is a slide that you probably all have a version of. It basically says it's much better to listen to the early signals for disease and catch it and treat it early than it is to wait for the time that it's shouting at you. And we quite intentionally have this figure that talks about the conversation of the body. So rather than listening to cancer shouting at you, I have stage 4 colorectal cancer, you really want to catch the whisper that says, I have an early adenoma, I probably should take it out. And so we think about that in this disease, we think about this in coronary artery disease, we think about these in other indications as well too. It's crowdsourcing health information, if you want to use a social media buzzword. But we think it actually applies here. So when I look at you and I want to listen to your conversation of the body, this is what I think of. So this is a picture of the proteomics picture I see when I look at a fraction of plasma. It's roughly, in this particular mass spec injection, about 50,000 molecular features. We typically collect about 300,000 molecular features from each patient. And we're looking for that pattern, that signal in the weeds that says you have one thing or you don't. It's the ultimate high-dimensional data problem. I'm actually a nucleic acids molecular biologist by training. I developed the SNP arrays for Affymetrix, so I know doing 2 million measurements on 100 patients is a difficult thing to do. But that's where Danny's expertise in information processing comes into play. So I love the idea of thinking about this being what happens in a physical when you go in the future. I'd like to say that the physical of the future will be that you go in and with 3 different receptacles, you'll provide urine, blood, and saliva. You'll sit there for a little while and the doctor will come in and give you a picture of your health status. That's the aspirational goal. That's sort of summarized in this particular slide to say where the status of all of our work is at the moment in this sphere. This has really been the past where a given lab might work on a signal transduction pathway for 20, 30 years, several research grants, wonderful papers, wonderful mechanistic work, and then they try and turn it into a therapy or a diagnostic tool. And it sometimes fails. And I came up in my career through Novartis and drug discovery where we cured diabetes in mice many times over, and then it doesn't work in people because guess what? This molecule that does this in muscle does that in the liver. Right? And so this very siloed way of thinking has been problematic. This is where we are now. So we have all these wonderful technologies to measure everything about you. I can deep sequence your blood, I can look at all your proteins, your metabolites, but the downstream healthcare system is still set up for just one test per indication. And so even though I can measure all of these things about you, I have to turn it into a single test so it fits onto a Roche box or some other lab-specific test box because the paradigm is for that. What we aspirationally believe is that eventually we're going to get to a world where this discovery technology is the test. If I measure everything about you quite easily, why not test for many things at one time? Hence my metaphor about testing all 3 things at one time. So this summarizes that again. Basically, we'll look for all the differences. That's what we would like to do. Now, what's the technology we use to do this? It is mass spectrometry. It is a wonderful technology for discovery and for targeted discovery as well as commercial development. We use it in 2 modes. My goal is to give you a little bit of an education about this particular mode. But in this discovery mode, like I said, we go and look at all the bits and pieces that are floating around. Hundreds of thousands of potential signals. I don't know what they are. I let the math tell me what's interesting, and then I go figure out what they are. So it's hypothesis-generating discovery. But it turns out that mass spec has been used for decades in the pharma industry for following small molecules around. Well, if you digest proteins, they become small molecules. Right? And so what we are now doing, and many other labs are doing, are using mass spec to multiplex measurements, where you basically have this wonderful box for doing the ultimate multiplex ELISA, and can look at many, many proteins at one time. That's great because then what you can do is you can take the entire body of literature of a given field, like colorectal cancer, and say there are 200, 300, 400 good proteins, not so good proteins that never work very well because people have worked on them one at a time. And then they failed. But if you can actually measure them all at one time and then go look and see whether you get combinatorial performance, that may be a huge win. And that's what this talk is going to be about, exploring this particular axis where we go and do this work. So I'm going to tell you a little bit about the work that we've done over the last 5 months. So none of this is in print yet, but it's pretty exciting, and we're getting ready to go now to some very exciting sample sets with Professor Brenner and others as well too. This is a description that just says, this all works because we treat it as an industrial process. So if I was going to walk you through the chemistry of discovery, I'd take you through panel by panel on this slide, but in the interest of time, I will not. I will speak a little bit to this slide. At least in the US, proteomics has been what we call a dirty word for a long time. It has not worked very well. People have spent a lot of money on some very big, expensive boxes, and it hasn't really generated much. That's probably because what they've done is they spent a lot of money on big, expensive boxes and not taking care of all the other parts of the experiment. So when we do a measurement, we think about all those other parts. This is, This is actually software that we have that real-time keeps track of all the processes. This is a bit of mass spec data. And this is a graph of all of the factors that went into generating that data. You have to think of it like an industrial process, even though you're doing academic research. And that's really what makes the whole thing work. It's a systems engineering approach, and that's Danny's expertise. So going forward, we get to highly reproducible data. I can take the same sample, run it with different people, different reagents, different days. I can superimpose that data off of the mass spec, red and green in these two particular samples. Yellow is where they overlap in position and intensity. Guess what? The slide is mostly yellow. That's good news. The intensities map as well too, so this tells us we can actually go and do this experiment. Or to put it another way, it used to be in this field, technical noise dominated, biological signal was way down here. And what we're doing now in the field, and what we're doing in particular, is driving the technical noise down to a point where it actually works. We, like many of you who are in this game of looking for signal, have a paradigm and a pipeline and software that we write to go and analyze and look for signals. I won't walk you through that. Suffice it to say it's fairly robust. We're constantly sending our results out to our collaborators and leaders in the field to make sure that we're not, as I say, chasing our own tail statistically. It's very easy to do that in a high-dimensional space. But we're deploying this now in this sort of broad approach towards getting something to market. So we're going to talk a little bit more about targeted mass spec and how it actually is involved not only in discovery but in commercial test development. We just said it all, I won't belabor it again, but for me coming to this field just since the past 2 years, after many years studying diabetes, cardiovascular disease, obesity, and other diseases of the immune system, you can prevent this one. You know, it's a no-brainer to go do that, and you're surprised why people don't go and have these screening tests. And in the US, it's terrible. It's worse in other places, but half of the people that know that they should go don't go. I'm 52, I finally went. I'm glad that I did. I'm healthy, that's good news. But the goal is to actually get that to happen. And unfortunately, most of the diagnostic tools that are available really catch the late stage things predominantly. The aspirational goal is to catch things early, precancerous state, eventually get to a pinprick. Imagine it's a pinprick that goes on a dried blood spot. You mail the card in, it goes on a mass spec, and I measure tens, hundreds of proteins at one time. And maybe not just for colorectal cancer, but for many other things at the same time. That's what we would like to do. This is a necessary evil slide. Good science is not necessarily good business nor good health practice. If I come up with a great test and it costs $100,000 to do, it's not going to help anybody because nobody's going to use it. Nobody's going to pay for it. So one of the very first things we do when we see an interesting area is we go and do the health economic study and ask, at what we think we can do this test for in the marketplace, will it add cost to the system or not? In this particular case, modeling a test at about $200 with acceptance rates for use, oops, sorry, wrong button again. of about 30%. This is a million patients over a 5-year period. We basically spend the same amount of money. It's that we've shifted some of this money from treating cancers to actually screening and preventing. And my argument, obviously, is that I'd rather spend dollars on preventing it than on treating somebody that has the late stage. So this actually worked out quite nicely. And it's a virtue of mass spec that I can measure 50 or 100 proteins at a very low cost. We have collected a number of our own samples. Prospective studies, this is a protocol for patients that are coming in for a colonoscopy. We get them diagnosis naive. We collect their plasma and then go and look at their markers to see if we can then build a model that predicts the presence of advanced adenoma or colorectal cancer. That's harder to do with cancer because the incidence is low. But we have a carefully controlled study. We keep track of all of the relevant features, pre-analytical variables, position, size, appearance of the various polyps, adenomas, pathology reports. All this is very important when you're looking for a pattern. You want to make sure that you're not chasing a bias in the study. Now, the way that we actually do targeted mass spec, and I want you to think about it because there are other experiments that we can do, you're basically taking a protein, digesting it to bits and putting it into the mass spec. So here's that big picture I showed you before. And what we do in the mass spec is pick one of these little things and go in like a particle physics experiment, break it apart into smaller fragments like this, pick the really bright and consistent ones and go and measure those. And this I can do on hundreds of patients, this I can do on hundreds of thousands of patients. And so we pick proteins that we want to go study and develop these assays. And for this particular study, that looked like this. So we combed the literature and came up with a list of 188 proteins that had what we felt was first-tier evidence. Publication, some involvement, predictive ability for cancer, almost all not very good. So they're considered interesting academic studies but fail commercially. There was presence in plasma or serum. And then we said, that's great. Let's go measure all of these at the same time from a large collection of samples. And the way that we prep that work then is we go and say, well, these are the protein names. Let's broaden it out so all the protein isoforms are there. There's a certain number of tryptic peptides because we digest proteins to peptides. And then we filter through to the ones that fly well in the mass spec, that have good homology so that they're separate from all the other proteins, and that actually quantitate very well. At the end of the day, in a single 30-minute run, I'm making 1,400 measurements, quantitative measurements on all these proteins. This is an example of what that actually looks like. We actually, for the targeted proteins, put an isotope-labeled reference peptide into the same sample. So half of those measurements are on the endogenous level and half are an internal control. So it's very good, reproducible data. And this is where we are now on adenoma. And I'll bet that everybody here has seen a rock plot, but there's always somebody that looks at it and goes, I don't know what the heck that picture is. So if you'll allow me just the 1 second. This is a measurement of how an assay is performed. This curve here. You want to push this curve into the upper left-hand corner. This is perfect, 100% sensitivity, 100% specificity, and this line is random. And this is where we are right now in a 128 sample study. That's half advanced, it's a case control study, half advanced adenoma, half control. Looking at just 4 proteins in a classifier. And what you see here on the plot are the FDA docket reports for the 2 recent conversations that were actually happening. One of these is the FIT test and the other is a company that I won't name. But at least in this cross-validation within the discovery group, we're exceeding the performance that we see, that has been seen commercially. So we're pretty excited about this ability to do that. On to CRC then. Here we have, and sorry, let me go back to this set. This sample set was our own very carefully collected prospective sample set. Easier to get to because advanced adenoma prevalence is much higher. Cancer is tougher, and we had to go and buy samples. I'll bet many of you have looked at these companies by name up here, Proteogenics, Astra, and Cap Bio. We bought samples from all these and pooled them together because they all have horrible individual biases for discovery. These are not great sample sets. But we just pooled them together and split them into discovery and validation. And then went and did the experiment that I described, looking at those 188 proteins along the way, holding one out as a discovery group and one as the validation group. And here's where we are in that study. So this is the discovery group. Again, 10 rounds of 10-fold cross-validation, AUC 0.82. And this is the performance in the validation holdout. AUC is slightly higher. It's just the luck of the draw of the way we divided the samples. What's interesting here and very instructive is that in the validation plot, all these little gray lines are the components for the 13 proteins that comprise this signature. It is probably going to be true in the future that good performing signatures will be this combinatorial benefit. Right? I think the age of one marker does it all has come and gone. It's like the monogenetic studies from the 60s, 70s, and 80s. We're now playing in this multivariate game. The math is tougher. Studies are tougher. I teach all my molecular biologists statistics now. They all learn R after they learn how to clone things. Just showing where that performance is with respect to the one blood test. Again, the FDA recent docket and the other FDA recent document for the stool test. So we're in the zone we'd like to be in. When we look at our cancer samples to see if there's a bias for stage or a bias for sites in terms of misclassification, no one thing is throwing it off, so we feel pretty good about that. So this tells me that I have a signature that I can then go play in some very valuable samples. And those samples are the ones that are prospectively collected, where they're not diagnosis aware. You know, if you're told you have cancer and you're going to go in for surgery and I take a plasma sample, I guarantee you did not play tennis the day before. You didn't ride your bicycle to work. You probably treated yourself differently. So all of those sample sets are a little bit suspect. But we want to go into some very carefully collected sample sets where we don't have those biases along the way. Now, I'm playing this combinatorial game, right? So I've measured 100 proteins in this particular set, and I've evaluated how well each possible pair of two of them work. And there's an interesting bit of data that comes out of this meta-analysis. And what you see here is that there's a non-random distribution about how they well in train versus test AUC. There's a group of proteins that are consistently in these high-performing classifiers. So mass spec, targeted mass spec, is a very useful way to take all the knowledge that's in a field, all the poor-performing players, go measure them all at once on one sample, and find out whether or not you get the big win when you go for this combinatorial performance benefit. And we're in the process of taking this through to commercialization. In the interest of time, I'll just conclude with this reminder. What I've just described to you is an interesting technology that allows one to capitalize on prior knowledge very rapidly. This whole study just took the past 5 months. And that's this thing, but this is what we eventually want to do. Danny Hillis is very fond of saying we diagnose cancer geographically. You have colon cancer, you have stomach cancer, you have pancreatic cancer. You don't call the plumber when you have a leak and say, I have kitchen water, please come and fix it. Or I have bathroom water, please come and fix it. You say, I have a broken pipe. There's probably a lot of mechanisms that are common to many things. There's probably, in an aspirational view, one test that allows us to catch many things at one time, and that's what we'd like to do. And on that note, I'll stop. Thank you very much for listening to me.
Thank you very much for sharing this first very exciting data on your new technologies. I'm sure there are some questions. Yep.
Obviously, it's amazing and fascinating, but can you share with us some of the data, it kind of theory? Any specific, what is the signatures of these molecules?
Right, and so this is where the disadvantage of being a commercial entity kind of intrudes.
This is academia here.
That's true, but it's a first-to-file world now, right? And so these data here are just a few weeks old, and we're in the process of preparing the patent applications. They are known proteins, right? I can tell you that if I showed you any of the proteins on this list, you'd go, oh, aha, because they all came from the literature associated with colorectal cancer. Right? None of these are new inventions in this particular study. We have a whole body of work where we have novel things that haven't been shown up before. But if I showed you this list of 13 proteins for the one or 4 for the other, you'd go, oh, okay, that makes sense. There's literature on that. The inventive part is that those 4 together work better than you would have expected differently. So I apologize. I understand your frustration. I've been on the other side of that quite frequently. We look forward to, at the appropriate meeting and in the appropriate publications, disclosing what these signatures actually are.
My question would be, would this test be specific for colorectal, or would it be possible that a positive test would just indicate if there is no colon tumor, that the tumor is somewhere else in the body? So my question would be, what would we do with individuals which have a positive test here, and then they receive colonoscopy or decolonography, whatever, and this is negative, but still there is a positive test? How would we deal with this issue? So would it kind of be a pan-cancer test then?
Yeah, and so it's the specificity question. This is where most tests fail, because people will focus, they'll do what we in the US call grind and bind experiments. They'll take one bit of tumor and grind it up and go look for all those things, and it turns out that that protein is expressed in 50 other tumors. In the adenoma data, we did it as an all-comer study. So everybody that came in, these several thousands of patients that we've collected, came in not healthy versus the presence of advanced adenoma. It was everything else in the intent to test population versus advanced adenoma. So to a certain extent, this early study already is a test for specificity in the general population. In the colorectal cancer samples, where they were collected basically from patients who were coming in for GI surgery, there the comparison, the case control was CRC GI-related surgery versus non-CRC GI-related surgery. So in a way, it's a tougher specificity test for GI-related issues, but not necessarily for cancer-related issues. And the gold standard for that, obviously, is to go into those prospectively collected sample sets from the intent to test population. But that's where you have to go and collect 5,000 or 10,000 patients and then go and test them. And our group, we're collecting from 3,000 patients right now prospectively. They'll be available by the end of the year. We have a wonderful relationship with Professor Brenner, and we'll have access, I think, to some of his blitz or doc samples to go and test there, as well as another group in the US. So we have that very much on our minds, because that's where most tests actually fail.
I have another question. You have shown your preliminary results that were almost equal, the performance for advanced adenomas and for colorectal cancer, which seems unusual on first view. Do you have, or would you have expected that, or do you have an explanation?
In other words, am I surprised that it worked so well? Is that, could you? No,
why the results for advanced adenomas, they seem to be so good compared to other blood tests, whereas for the colorectal cancer.
Yeah, I think this is the nature of using this, if you'll forgive me for dropping back to the metaphor, the conversation of the body again. I think most of the existing tests are trying to catch either physical lesions or abrasions of tumors, or they're trying to catch late-stage molecular events, and that's why they don't work early. When I go and listen to all of these signals, the sort of little bit of input, it could be that I'm picking up either, I'm now sensitive enough for the early stage, or what we're detecting is not a signal from the adenoma itself, but from a fertile field, if you will, something that is prepped for the presence of adenoma. Remember, I cannot say that the signal specifically came from the adenoma. It could be that it's just from the intestinal wall itself. The colon wall.
Okay.
Thank you very much.
We have